Hello everyone. Today I'm going to talk about the glomerular diseases in detail. Then I'll be talking about the nephritic syndrome. So what I will be speaking about today is the overview I'm going to give you in this slide. That is the recap of yesterday's class, then tendencies of glomerular diseases, then microscopic features, nephritic syndrome, nephrotic syndrome, and its pathogenesis. So now, coming to the basic glomerular structure, I told you the glomerulus is a duct of capillaries that is formed by the afferent and efferent arterioles invested in two layers of epithelium, that is the visceral epithelium and the parietal epithelium, right? So the filtration of the glomerulus is done by the endothelium, that is usually fenestrated, that is 70 to 100 nanometers. Fenestrations are seen here. Then coming to the mesangium, which is the supporting structure for all the capillaries which are formed in the tuft, that is a mesangial matrix that supports it in the mesangial cell. The mesangial cells are nothing but they are the phagocytic cells. They kind of proliferate in any kind of glomerular injury and then will subsequently help in the fighting the disease, and is a part of pathogenesis of any glomerular disease. Then coming to the visceral epithelial cells or the podocytes. See, the visceral epithelial cells are the ones which are called as the podocytes. They are having the filtration slit. The filtration slit integrity is maintained by the nephrin, which is an extracellular molecule that is held together by the intracellular podosin and CD A2B protein and also the actin filaments. So any disturbance in the structure of nephrin or podosin will lead to glomerular pathology. Okay, and then coming to the parietal epithelium. So you know that there are two epithelial layers, that is a visceral epithelial layer and the parietal epithelial layer. This parietal epithelium will get proliferated and helps in the formation of crescents in severe glomerular injury, where there is accumulation of these parietal cells. Okay, that's how the crescents are formed. Now, I told you that there are some things which help in the filtration. So what are those? The filtration in the glomerulus happens depending upon two things. One is charge dependent filtration. The other one is size dependent filtration. Usually highly ionic molecules like albumin are not crossing the glomerular basement membrane, right? So according to this, the deposits are being taken place. And the second one is size dependent. High molecular weight molecules cannot pass the glomerular basement membrane. So these are the things you have to know. In the structure of the glomerulus, we also should deal about the glomerular basement membrane, the central dense, electron dense layer, the lamina rara externa and lamina rara interna, which are the electrolucent layers. Here you can see this is the blood and these are the epithelial food processes. Okay, in this, the structure of the basement membrane is important because it is formed by the collagen molecules which are called for A1 to called for A6, that is the alpha chains. Okay, so alpha 1 to alpha 6. So these chains are having a C terminal and an N terminal. Okay, in between there is a triple helical substance. Okay, these all together are bound to form the basement of the glomerular structure. So because of this, the structure of the basement membrane is important because there is this non-collagenous domain 1 at the C terminus will be a crucial factor because the antibodies may recognize the non-collagenous domain 1 and then there can be an antigen-antibody reaction taking place, and because of that there will be a disease. So this can be renal limited or it can also be identifying the same basement membrane molecules which are present in the lung alveoli and lead to good pastures, right? So we have spoken about the pathogenesis or the response to the glomerular injury. So what happens when there is a glomerular injury? We know that there are few cells in the glomerulus now, right? The epithelial cells, the endothelial cells, the parietal visceral epithelium or whatever. So in response to any glomerular injury, the first thing that happens is hypercellularity. Hypercellularity in terms of either proliferation of mesangial cells, leukocytes, endothelial cells. So what happens when something proliferates? Obviously, it tends to accumulate. So because of this accumulation, it can cause a glomerular pathology. Okay, thereby the GFR will decrease. So that you remember, the first pathological response to the glomerular injury is hypercellularity. And the second one, which is important in the response of the glomerular injury, is that the basement membrane thickening. The basement membrane thickening happens as a duplication. Okay, there is already a basement membrane, there is a formation of another basement membrane, that is a basement membrane thickening, or a duplication, or there can be increase in the protein synthesis as seen in the diabetes nephropathy. So we are seeing the first pathological response as hypercellularity in terms of different cells which get proliferated and they kind of release cytokines and chemokines and the soluble mediators. Then the second one is the basement membrane. Either it gets thickened, or there is increased protein synthesis, or there is duplication. And the third one is hyalinosis. The hyalinosis is nothing but there is a protein which is insulated usually in the renal parenchyma, and when this is being released, that is extracellular eosinophilic substance, if this is amorphous substance, if this gets released, then it can obliterate lumen of the capillaries and then cause a glomerular pathology. So this is hyalinosis. The other one is sclerosis. Sclerosis is extracellular collagen matrix deposition. Okay, so these four things are happening in response to any glomerular injury. Hypercellularity, bone marrow thickening, or whatever, thickening, duplication, or increased protein synthesis, hyalinosis, and sclerosis. Remember, these four are happening, right? So now coming to what happened, where this can happen. Like they can happen either diffusely, that is the entire renal parenchyma is involved, or it can occur focally, that is fraction of the glomeruli in the kidney can be involved, or global, that means the entirety of an individual glomerulus is involved, or segmental, when a part of individual glomerulus is involved. So remember these terminologies which are very, very important, right? So these are the histological changes which we express in the glomerular disease, right? Either it can be diffuse, focal, global, segmental. Other than this, there can be the capillary or mesangial things which can occur. We just remember these terminologies which are very important, right? So after that, now we have come to a place where there is glomerular injury, there is response to the glomerular injury, and in response to the glomerular injury, we also know that there is proliferation of certain cells, certain soluble mediators like eicosanoids, chemokines, and cytokines that are trying to recruit more leukocytes, and then they are causing the injury, right? And beyond that, there is something called as the immune-mediated injury that is happening. The immune-mediated injury, I told you, will happen in three ways. One is antibody-mediated, the other one is alternate complement activity mediated, and the third one is cell-mediated, right? So now first coming to the antibody-mediated, we have seen that it can be in situ with a fixed antigen or a planted antigen, or it can be a circulating immune complex, right? That means in situ binding of, there is an antibody that is getting attached to the antigen which is recognized in the form of a porocyte, that is a visceral epithelial cell in the glomerulus, or the glomerular basement membrane, right? So there is already the antigen recognized in the form of the glomerular basement membrane or the visceral epithelial cells. And to this, if it gets attached, now this is called as the in situ binding. Okay. Then here there can be some planted antigens. When we talk about the planted antigens, we gave an example yesterday as infants fed by the cow's milk. The milk antigens are absorbed from the gut and they act as substrate for the formation of antigen-antibody complex. As they get planted on the glomerular basement membrane, they get recognized by the antibody and then there is an immune complex formation. So that is the second type. So in the in situ type, we have either fixed antigens or planted antigen to which the antibody reacts. The third component, the second component, that is in situ, we have planted and fixed, and the second one is the circulating immune complex. The circulating immune complexes can be formed either as a result of an endogenous antigen or an exogenous antigen, right? So the endogenous antigen, we know that the best examples are the lupus nephritis and the IgA nephropathy, right? The exogenous, the antigens can be instead by number of viruses or bacteria or their products. So here we can see this diagram depicting the deposition of immune complexes, which will result in two types of immunofluorescence. That is either granular immunofluorescence or a linear type of immunofluorescence. Linear type of immunofluorescence is only one, that is anti-glomerular basement membrane antibody, which will cause an disease in the form of linear immunofluorescence. And in circulating immunocomplexes and the podocyte injury, we have this called as a granular immunofluorescence. Okay, just remember that. So now, this is a classification of immune-mediated glomerular injury, that is antibody in situ, which can be a fixed intrinsic tissue antigen or a planted tissue antigen, right? So here we have non-collagenous domain 1 of type 4 collagen antigen and phospholipase A2 receptor antigen as detected in the membranous glomulopathy and the mesangial antigens. So here we have circulating immune complex deposition by the endogenous antigens and exogenous antigens. So the other type is a cell-mediated injury, and the other one is the activation of alternate complement pathway. So here we spoke about the most important in situ immune complex deposition experimental model known as a Heyman nephropathy, right? That is caused when the antigen is produced in the rat and in the form of megalin that will initiate the pathology, right? So what is happening there is that there is an identification of the membranous nephropathy that is happening as a result of the proliferation of the cells that are getting deposited subepithelially. Okay, so what is happening here in humans rather than experimental model in the body is that the antigen that is recognized in the humans is the M-type phospholipase A2 receptor antigen. So because of this, there will be a reaction that is taking place. So because of that, there will be proliferation of cells that are getting deposited subepithelially and they are forming the membranous glomerulopathy, right? So the other one is a non-collagenous domain 1 of type 4 antigen. We already spoke about the anti-GBM nephritis, right? So the planted antigens can be exogenous or endogenous. We spoke about the infant cow milk and all. The circulating immune complex depositions as endogenous antigens. So now coming to the cell-mediated injury. The cell-mediated injury is usually happened by the sensitized T cells which act in coordination with the antibodies and then they kind of help in the pathology of the glomerular injury. So the other one is activation of alternate complement pathway. The activation of alternate complement pathway, you find it in the dense deposit disease or membranoproliferative glomerulonephritis type 2, wherein we'll be discussing about that in detail while we are discussing each individual nephropathy. Okay, so before going to the actual topic of today, that is tendencies, how they manifest and all, I would also like to tell you that there will be some kind of progression that can happen once there is a renal glomerular injury. How this progression is happening? We spoke about that yesterday in form of two things, that is focal segmental glomerular sclerosis and the other one is tubulointerstitial disease. Focal segmental, focal is a fraction of glomeruli involved, segmental, focal is a fraction of glomeruli involved, segmental, a part of each glomerulus that is involved will cause a localized reaction. So because of that, as the other glomeruli, other people in the family will try to expand, that is the glomerulus will get hypertrophied and then there can be increased intraglomerular hypertension, intraglomerular pressure will, intracapillary pressure will get increased, and because of that there can be systemic hypertension in the patient, and then finally the glomerular injury will get accentuated. Then the other one is a tubulointerstitial disease. The tubulointerstitial disease can be a result of a direct injury of the toxin or it can be related to the glomerular injury. Okay. So because of this, what happens is that it will also add to the existing glomerular pathology. See, once if there is a disease, a glomerular pathology, which is decreasing the GFR by 30 to 50%, then forget about the inciting agent or forget about the disease, it can progress to end-stage renal disease irrespective of the inciting agent and the disease which is initially there. You're getting my point? Initially, if the disease, if the glomerular injury has already produced a 30 to 50% of reduction in the actual GFR, then what happens is that this disease will progress to end-stage renal disease steadily irrespective of the initial inciting agent. For example, a bacteria, virus, the immune complex or whatever that is inciting the reaction. Now, in spite of that, if you remove that also, the disease will progress steadily, right? That is a very, very important point you have to remember. And we also spoke about different deposits. See, the epimembranous deposits are seen in membranous nephropathy. The subendothelial deposits are seen in membranoproliferative glomerulonephritis. Mesangial deposits are seen in IgA nephropathy. The subepithelial humps are seen in acute glomerulonephritis associated with streptococcus. Okay, these things where the disease occurring, why? If you want to remember that, you have to get a concept that highly cationic molecules cross the glomerular basement membrane and then they find subepithelial location. The endothelial location is usually for those anionic molecules which cannot cross the glomerular basement membrane. And the neutral ones are usually seen in the mesangial. The mesangial cells, they are getting deposited in the mesangial. Remember the concept, you just know whichever question, in whichever way it's given in your essay or in any kind of exam, or you actually find a patient for yourself, then you will know that where the disease pathology can be, whether it is subendothelial, subepithelial or mesangial or related to what. Okay, so that is about it. Usually when there is an epithelial antigen antibody reaction, there will be this proliferation of cells, I told you. These proliferation of cells and this antigen antibody reaction together will form a deposit or shredded as a deposit at different places. So the localization of these deposits is determining the progression of the disease. That's number one. Okay, you have to remember the localization where it is happening. The second major point is that if you know that there are some deposits, these get degraded. Okay, eventually, for example, in Poststreptococcal Glomerulonephritis, these things get degraded because you can actually cure. It is reversible. You know, you're kind of treating the streptococcal antigen streptococcal disease as such in toto as a systemic disease. Then you are trying to eliminate the inciting agent. Then these immune complexes and these depositions can get degraded, right? So, or if they are not getting degraded and there is a continuous inciting agent, for example, like in lupus nephritis where there is a DNA or having the being the inciting agent, then there will be a prolonged disease and a more chronic progressing glomerulonephritis that can happen. So you have to remember that the glomerular structure is important, the response, hypercellularity, glomerular basement membrane thickening, hyalinosis, sclerosis, then the immune-mediated injury in form of in situ or circulating immune complexes or T-cell mediated or alternate complement mediated. And once there are deposits, the deposits localization is important. They can be shredded off and then they can be degraded because of the degradation. If it is not happening and then it is continuously, there is an inciting agent, there can be a progression to a more severe disease or more severe illness. Once this disease is formed, it is actually progressing to end-stage renal disease with the starting of focal segmental glomerular sclerosis and tubulointerstitial injury. And after that, there will be progression to the end-stage renal disease, irrespective of the inciting agent or the disease which is initially present, and the rate is usually steady. Okay. And here you have to remember that there can be coagulation factors also in response to the injury with the hypercellularity and the proliferation of all the cells. And these coagulation factors will kind of make the body react, and you know, the person actually die with a myocardial infarction rather than the renal injury itself. So this is about it. I told you another term called as podocytopathy, which is involving the visceral epithelial cells, wherein you find the effacement of the podocytes and then degradation, that is detachment of the podocyte, and that may cause the plasma protein leak. Okay, just remember these terminologies are important to know the actual disease process. So now we'll see the tendencies of glomerular diseases, how they manifest in nephrotic and nephritic features. Nephrotic is more than 3.5 grams proteinuria per 24 hours. Then there is hyperlipidemia, lipiduria, severe edema, hyperalbuminemia, right? Then nephritic features. See, these are the diseases. I told you the glomerular diseases are primary or secondary. You know, the secondary diseases are usually associated with systemic involvement. The primary diseases are these things. And the third one is also there, wherein you find some hereditary disease. So here you see the nephrotic proteinuria is seen in minimal chain glomerulopathy, membranous glomerulopathy. FSGS, it is usually associated with nephrotic range, and in minority of cases, it is that, you know, take this plus as minority and this as majority of the cases. And then the Cresentric Glomerulonephritis, where nephritic range proteinuria is predominant. Now, what are the light microscopy features? I told you there will be hypercellularity, so that you can find in the microscope, under the microscopy while you are examining. Then you find infiltration by the leukocytes in form of neutrophils, monocytes, and macrophages. Proliferation of endocapillary cells, proliferation of extracapillary cells, that is the epithelial cells. Usually the prisons are formed by the proliferation or accumulation of the parietal epithelial cells. More, remember crescent formation, parietal epithelial cells. Then increased extracellular material, sclerosis, localization of immune complexes, where they are subepithelial, subendothelial, or mesangial, increase in the collagenous matrix, increase in plasma proteins, fibrinoid necrosis, deposition of amyloid, right? So, immunofluorescence features. Linear staining of glomerular basement membrane, I told you, will be seen in anti-GBM antibodies. Then multiple plasma proteins associated with diabetic glomerular sclerosis. Monoclonal light chains usually is in overflow proteinuria, usually in multiple myeloma. The granular immunocomplex staining in the mesangium, IgA nephropathy, capillary wall, membranous glomerulopathy, mesangium and capillary wall lupus glomerulonephritis. Okay, irregular fluffy staining is usually seen in AL amyloidosis and AA amyloidosis. See, you know that some light chains are getting deposited in the amyloidosis, right? So these light chains, if it is ALL or AA, they're usually irregular and they have a fluffy stain. The electron microscopic features include the electrons dense immune complex deposits. I told you mesangium, IgA nephropathy, subendothelial, lupus glomerulonephritis, subepithelial. The classical example I gave you as hematonephritis. Remember that there is a subepithelial location, right? Then glomerular basement membrane thickening in diabetes glomerulosclerosis, replication or duplication that is happening in membranoproliferative glomerulonephritis, collagenous matrix expansion that is FSGS or focal segmental glomerulosclerosis, and fibrillar deposits as in amyloidosis. You need not by heart all these things, okay? You just remember the concept of the glomerular injury and the glomerular pathogenesis, then you just get where they get deposited, highly cationic, anionic or neutral. Remember that. And what is the substance that is getting deposited and how is the light microscopy. There is only one thing in the immunofluorescence which is giving the linear thing, that is the anti-glomerular basement membrane. No confusion at all. So now coming to the nephritic syndrome, we have finished the pathogenesis, the immune basis of the glomerular injury and various glomerular diseases and their classification as primary, secondary and hereditary. We will go in detail of each glomerular disease. Before that, we'll see about the nephritic syndrome, right? So nephritic syndrome is characterized by hematuria. Either you can just see the urine collected urine as red color or pink color, if there is hemoglobin, or red color, if there are multiple RBC, then it can be visible grossly or you can see under the microscope, right? Variable degrees of proteinuria, but not in the range of nephrotic, that is not more than 3.5 grams per 24 hours, but more than 150 milligrams per 24 hours. Remember that. And decreased glomerular filtration rate. Okay, obviously GFR gets decreased. In what way you will find, what are the investigations you'll ask for? Serum creatinine, urea, blood urea, nitrogen. You will calculate the creatinine clearance. There is blood urea, nitrogen and creatinine ratio that will take. Then you do this renal function test and associated, you will try to find if there is a hematuria, you will try to find the cause for it, ultrasound and all the imaging you do, and then you get to a first basic test before all procedure is the complete urine examination, right? So elevated blood urea, nitrogen and serum creatinine, oliguria, saltwater retention, edema and hypertension. So what are the types? Acute glomerular nephritis, which is rapid and reversible, and the other one is rapid progressive glomerular nephritis, which may resolve with treatment. And both of them occur from, the glomerular injury can occur from hours to days. Remember that. And coming to this, the acute glomerular nephritis is acute glomerular nephritis. That is a cluster of diseases characterized by diffuse proliferation of glomerular cells associated with influx of leukocytes caused by the immune complexes. You know that whenever there is an infection, what happens is that the soldiers of a body, that are the leukocytes, will get into action, and then there is diffuse proliferation of the glomerular cells itself, the visceral epithelial cells, parietal epithelial cells, endothelial cells, mesangial cells, all these things will get into the action. Okay. Now coming to poststreptococcal glomerulonephritis, usually it is decreasing in frequency nowadays because anybody who is getting sore throat, they kind of go for the treatment. Because of that, the sore throat, the streptococcal antibodies are already present and they're not forming any kind of, they're not doing damage to the kidney, and prior to that itself they're getting treated. So what happens is that 1 to 4 weeks after a streptococcal infection or a skin impetigo, it is usually seen, this kind of glomerulonephritis. The children, usually 6 to 10 years of age, are getting affected. Usually they are of low socioeconomic status, and then there is they are situated in a place where there is excessive crowding. So there is immune complexes contain streptococcal antigen and specific antibodies which are found inside. Okay, just remember that there should be an antigen, right, for the reaction of the antibody and immune complex formation. That is usually taking place in situ. And then it is usually the A beta hemolytic streptococci that is usually implicated in this. And then the strains types are 12, 4, and 1. Okay, the M protein of the bacterial walls is the inciting factor. See, either the virus or the bacteria itself can cause a disease or its product. Okay, for example, in tuberculosis, you know, the cell wall is important, and here the M protein of the bacterial cell wall is important. Okay, so something like that which is inciting the inflammation or immune reaction. So the latent period between the infection onset of nephritis is compatible with the time required for the production of antibodies. So the latent period of the infection and the formation of antibodies is compatible. The elevated titers of see how we know that the person is having glomerulonephritis by history, by their the economic status and all. You just take the history, the sore throat history. Then if you have an investigation, there will be elevated titers of antibodies. Okay, and then second one is serum complement levels are low. The complements, why they are low? Because the complements get activated here in response to the injury which is caused, and then the complements get consumed, so there is consumption of the complements. Then because of that, the serum level of complement will be low, and there will be some complement deposition also taking place. So because of that, the complement levels are low. Pyogenic exotoxin B is usually the principal antigenic determinant in most of the cases, but not all the cases, right? Then the hump-like deposits are characteristic of this disease. The hump-like deposits, I told you, occur, see, whenever there is this bacteria itself is causing the disease, the inciting agent, then there will be the subepithelial humps. The subepithelial humps are there, but the bacterial products may go cross the glomerular basement membrane, or they may not cross the glomerular basement membrane and form subendothelial deposits. So the antigen of the bacteria which is causing the disease is placed subepithelial humps, and the other ones which are the bacterial products, they are forming the subendothelial deposits. Usually these subendothelial deposits are seen in the earlier days of the infection. So coming to the histology, the hypercellularity, see again, if you remember those 4 things which are same, you can remember any disease by that. So here the hypercellularity caused by leukocytes, both neutrophils, monocytes, proliferation of endothelial cells, mesangial cells. Severe cases, crescent formation. Crescent formation is by parietal epithelial cells. So this injury can be global or diffuse. Diffuse, I told you, the whole of the renal parenchyma is involved. Global, that is the total entirety of individual glomerulus is involved. The other one is swelling and leukocyte infiltration obliterates the capillary lumens. Once the capillary lumen is obliterated, there will be interstitial edema as well as a tubular injury happening because of this. So remember, the cells which are getting increased in number as a result of the injury, that is a response to the injury, where the localization, where they are, and then what happens as a result of that. Okay, so here we have this is a normal glomerulus, you can see. Okay, And here you can see this is the hypertrophied glomerulus because of proliferating glomerular cells and presence of numerous leukocytes in here. So you can see the normal glomerulus, and this one is the hypertrophic glomerulus with proliferating glomerular cells and leukocytes. So here this is an electron microscopy picture. You are seeing these are the visceral epithelial cells on the podocytes, and this is the subepithelial hump that is seen in the acute glomerular nephritis. And this is the granular staining which you can see as a result of the complement deposition. Okay. So what is the clinical course? There is malaise, the color-colored urine 1 to 2 weeks after the sore throat. After the sore throat is important. Then if you do the urine examination, there will be dysmorphic red cells, periorbital edema, mild to moderate hypertension. You know, these things happen. In the adults, what happens is that there is sudden hypertension or edema, elevation of anti-streptococcal antibody titers, decline in the C3 levels, which are seen in both. So elevation of anti-streptococcal antibody titers is important. Take it. Then minority of the children do not improve. Usually this is a reversible disease. Minority of the children will not improve if they are very much dehydrated. So because of that, there will be rapidly progressive form of glomerulonephritis. Acute glomerulonephritis, one is streptococcal glomerulonephritis. The other one is non-streptococcal glomerulonephritis, which we'll be dealing here. The streptococcal glomerulonephritis, post sore throat and all. Here we can see that non-streptococcal glomerulonephritis as a result of bacterial, viral, and parasitic infections, right? So this type of post-infectious glomerulonephritis differs from produced due to the staph infection, differs by the immunodeposits, which are IgA rather than immunoglobulin G. Remember that. IgA rather than immunoglobulin G. So the third one, that is after acute streptococcal, acute non-streptococcal or post-infectious, we came to the end stage of the acute glomerulonephritis, that is rapid progressive glomerulonephritis, usually a severe form of disease, progresses within hours to days, and this does not denote a specific etiological form of glomerulonephritis. That means it is not saying that the RPGN is not giving you a clue about whether it is streptococcal, non-streptococcal. Anything may lead to, you know, the final thing, the final result of all the type of nephritis is RPGN if not treated. So, severe oligurean signs of nephritic syndrome will be present. So, what are they? Cresentric glomerulonephritis, proliferation of parietal epithelial cells, along with infiltration of monocytes and macrophages. Parietal epithelial cells. So the rapidly progressive glomerulonephritis can be renal limited. That means there are three types. Okay, three types. Type 1, anti-glomerular basement membrane antibody. Type 2, immune complex. Type 3, Pauci-immune. Type 1 is renal limited. That means it is just the disease. The antibodies are identifying the glomerular basement membrane as an antigen. They are just limited to the kidney. Good pastures, I told you, they also identify the lung alveolar basement membrane. They react with it. They form the disease that's called as good pastures. Type 2 is post-infectious lupus, noxtagin purpura, and IgA nephropathy. Okay. Then type 3 is usually Pauci-immune, that is ANCA-associated idiopathic. Usually Pauci-immune is associated with vasculitis, right? Excuse me. So here we are seeing a depiction of a diagram of anti-glomerular basement glomerulonephritis. Here you can see that this glomerular basement membrane is identified as an antigen itself, and then the antibodies reacting to it. you can know that there is recruitment of more neutrophils by activation of Fc receptor, and then you can see that the complement is also activated here. So this type is either renal limited or good pastures. So they are characterized by this, and then I have already told you the treatment is plasma pheresis. For this, the patients recover with this. You know, I already told you about what is the. The second one is immune complex glomerulonephritis. It can be lupus, IgA, you know, nephritis, nephropathy. So here we are seeing the immune complex which are formed as a result of circulating or in situ, you know, the antigen antibody which are already there, they get react and deposited here. So you are seeing the antigen antibody complexes, they are activating the complement pathway also. So here we see this is a diagram representing the immune complex glomerulonephritis. The third one is Pauci-immune. Pauci-immune is they are usually associated with vasculitis, and then they are defined by the lack of detectable anti-GBM antibodies or immune complexes, either by immunofluorescence or electron microscopy. So we are not finding any antibodies or immune complexes. Then we have to think about ANCAs. What are ANCAs? It can be a P ANCA or a C ANCA, right? ANCA is anti-neutrophilic cytoplasmic antibody. You know that neutrophil has a cytoplasm and 3-lobed nucleus, right? So, anti-nuclear cytoplasmic antibody that can have two types of staining pattern, and because of this staining pattern, it is called as P ANCA or C ANCA, which are implicated in different types of vasculitis. The P ANCA is showing the perinuclear staining, and the C ANCA is showing the cytoplasmic staining. Okay, this is the diagnostic marker. So, how is the ANCA glomerulonephritis? I told you there is no antigen antibody we are seeing here, either subepithelially or subendothelially, no glomerular basement membrane antibodies are seen. Then how it is causing the disease? So, there is a neutrophil. The neutrophil is having this Fc receptors, and then this ANCA is going and binding to the antigens which are finding here either in the cytoplasm or perinuclear. So this was causing the injury. This anti-neutrophilic cytoplasmic antibody is causing the glomerulonephritis. Then it is called as Pauci-immune type of nephritis. So here, this is a classification to know what kind of rapidly progressive glomerulonephritis is defined by 3 types. One is anti-GBM glomerulonephritis, that is either associated with lung hemorrhage, good pastures. Without lung hemorrhage, it is just renal limited. The immune complex nephritis, IgA dominant without vasculitis, it is IgA nephropathy. IgA dominant with including systemic vasculitis, it is Knox, Conley, and purpura. SLE, you know, the inciting agent is usually endogenous DNA, then, you know, the fragments of it is causing lupus nephritis. Immune complex formed by the acute streptococcal infection known as acute poststreptococcal glomerulonephritis, which we have just discussed. Thick capillary walls and endocapillary hypercellularity seen in acute poststreptococcal glomerulonephritis. Subepithelial dense deposits are seen in membranes, glomerulopathy, others, and many others. So, we have rapidly progressive glomerulonephritis does not define a disease, but it is an end form of all the nephritis which are related to either glomerular basement membrane, either immunocomplex or Pauci-immune, which is called as ANCA glomerulonephritis. The ANCA glomerulonephritis, if is associated without lung hemorrhage, it is ANCA glomerulonephritis. Vasculitis with no asthma, no granulomas, it is called as microscopic polyangiitis. Remember, vasculitis, microscopic polyangiitis without asthma. Granulomas, no asthma, it is vagus. granulomatosis, granuloma, granulomatosis. Okay. Then eosinophilia, asthma, and granulomas, all three are present. Associated granulomas associated with asthma and eosinophilia, Chuck-Strauss syndrome. So I have touched about the nephritic syndrome in terms of acute glomerulonephritis, infectious, post-infectious, that is the streptococcal, non-streptococcal, and rapidly progressive glomerulonephritis types, anti-GBM, immune complex, and Naga glomerulonephritis. I have given the whole types here, where if you have a question that is saying that there is a lung hemorrhage, there is an anti-GBM antibody, then you know that you are leading to the answer as good pastures. You know that there is IgA dominant and systemic vasculitis, then you get to know that you have been leading to the answer as Synopsis purpura. Remember this table to know about the rapidly progressive glomerulonephritis. Now, how about the morphology? The morphology is that the kidneys are enlarged, there can be petechial hemorrhages on the cortical surfaces, there is focal and segmental necrosis, diffuse or focal endothelial proliferation, mesangial proliferation. See, proliferation is something which is happening in response to the glomerular injury itself. Okay, the deposition, and after that what happens is usually a result, the necrosis is a result of the inflammatory injury. And the hemorrhage is usually as a result of the thing which is the vasculitis. So the crescents are formed by proliferation of parietal cells, migration of monocytes, macrophages into urinary space. What are accompanying? Neutrophils and lymphocytes are present. Okay, so they obliterate the urinary space, compress the glomerular. Fibrin stands are usually frequently prominent in the cellular layers of the crescents. So here you can see this periodic acid shift stain. Here you can see this is the glomerular basement membrane, the glomerulus. Here you can see this is a crescent. The crescent shape, the half moon shaped one which is present here. This is the proliferation of the parietal epithelial cells, can be associated with neutrophils. You can see this is a neutrophil here. Again, here you can see this is the glomerulus, the basement membrane. Here you are seeing the parietal epithelial cell proliferation here in the form of a crescent. So that's how you identify the Cresentric glomerulonephritis under the microscope. So by immunofluorescence, immune complex mediated cases show granular deposits, good pastures show linear deposits, right? Pauci-immune cases have no or little immune reactants. So Pauci-immune is usually anti-nuclear cytoplasmic antibodies, can be P or C, right? So the clinical cause is hematuria with red blood cells, moderate proteinuria. Occasionally, if it is severe, can reach the nephrotic range, variable hypertension, edema. So what we have to analyze for investigations other than microscopy, it is anti-GBM antibodies, anti-nuclear antibodies, and ANCAs. So the recovery of renal function can be seen by plasmapheresis in good pastures. Here this therapy can be both used for pulmonary hemorrhage and renal failure. The other forms also respond well to the steroids and cytotoxic agents. So we finished the nephritic syndrome. Now just a little introduction about the nephrotic syndrome and I'll stop. Nephrotic syndrome, I told you, loss of 3.5 gram per 24 hours, hypoalbuminemia, then generalized edema, hyperlipidemia, and lipiduria. 5 things you have to remember always. And what happens here is the pathogenesis. There is a glomerular damage, the increased permeability of glomerular capillaries to protein, that is, there is plasma protein leak because of the glomerular damage. Because of the plasma protein leak, what happens? There is proteinuria, hypoproteinemia, and whenever there is hypoproteinemia, what happens? Decreased plasma oncotic pressure. You know that the oncotic pressure, hydrostatic pressure will be maintained in the blood vessel, right? So this, the blood flow itself is maintained by these two. The plasma oncotic pressure is decreased, so the fluid escapes into tissues causing edema. Because the plasma oncotic pressure is decreased, there is no proper volume. So because there is decreased volume, there is decreased GFR, increased aldosterone secretion, and there is fluid retention, and again it leads to edema. So now the compensatory synthesis of proteins by liver happens because of hyperproteinemia. This is the result of hyperlipidemia. So initially there is a glomerular damage and proteinuria obviously as a result of the glomerular damage. Because the glomerulus is damaged, the GFR is decreased and there is plasma protein leak, and that is causing hyperproteinemia. And hyperproteinemia, the proteins are the ones which are maintaining the oncotic pressure. The oncotic pressure as a result, if it is decreased, the fluid escapes into the tissue and there is hypovolemia, and all these things will cause increased aldosterone secretion and edema. And then there will be compensatory protein synthesis to compensate the hyperproteinemia which is there, and this will result in hyperlipidemia. So this is about the pathogenesis of nephrotic syndrome in very brief. In the next Monday class, I'll be continuing the nephrotic syndrome. Thank you very much for your patient listening. I want all you guys to just see the concepts, how beautiful it is about the glomerular injury and the mechanisms. You'll never go wrong in your life regarding the patients or anything regarding the renal pathology. Thank you.